as soon as a small seed is formed, it's believed that this can lead to fast aggregation process and exponential growth. And one of the reasons why the seed can be formed can be, for example, protein overexpression, such in our mouse model for amyloidosis, which I tell you later a little bit. So, as I said, this is an equilibrium, and therefore this is really challenging. This step is really challenging to study in vivo. But the idea is basically that by targeting this specific step, that we could delay this aggregation process and thereby delaying the disease onset, as shown with this dashed line. So in the present study, we asked the question, can we target the first aggregation events in vivo at pre-amyloid phase? And therefore, we used a very well-characterized mouse model, the APP23 mouse model, which overexpressed a human APP containing the Swedish mutation. And the first deposition in this mouse model was shown histologically at around 8 months of age, like this. And here you can see the increase of brain A-beta in this mouse model. We also measured brain A-beta levels earlier than the plaque deposition, and this is shown in this small box. And here you can see that from 2 to 6 months, there is basically no change, and then between 6 to 8 months, we have a slight increase. So therefore, we decided to start treatment initiation at 6 months of age. So before any detectable changes biochemically or histologically occur in A-beta. So we set up the time point. The next problem to address was that the first in vivo formed A-beta assembly, which has pathological relevance, is biochemically uncharacterized. So this basically means we don't know exactly what it is, which makes it hard to specifically target. And to overcome this problem, we used a set of different antibodies shown in this table, which target different epitopes of A-beta. And here you can also see that some of these antibodies were used in clinical studies. In this case, we used a murine version, as our disease model is also murine. And at this point, I would like to mention that I have a biochemically background and I'm not involved in any clinical studies. So I would consider these antibodies rather as a tool that we used. So we inoculated APP23 mice at the age of 6 months on 5 consecutive days with an antibody in the box up here. And then we waited 1.5 months to the age of 7.5 before analyzing these animals. And the first thing that we did, we wanted to examine the brain A-beta load after immunization. And for this, we used the brains of the treated animals, homogenated, and then used a two-step extraction method. So first with Triton and then with formic acid. So here are the results from the Triton extraction, and you can see on the left side, it's A-beta40, and the right side, it's A-beta42. And you can easily see that there is no obvious decrease in A-beta40 or A-beta42 with in animals treated with amyloid targeting antibodies. In the formic acid fraction, where it's considered to see all the aggregated A-beta, we see a slight reduction in the animals treated with ADO in A-beta40, which was shown to be significant when we measured A-beta42. So apparently ADO was the only antibody which was able to reduce something at this early age seen by Triton and formic acid extraction. So and this was the first hint that we were able to target A-beta assemblies at this very early time point. But we did not know if this had any biological relevance. So we used the brain homogenate from the treated animals, ADO in control, and injected it into the hippocampus of APP23 host mice. So this is a very established method to test for biological active A-beta. And in fact, we could see, so here you can see the hippocampus injected with ADO treated homogenate, and the other one was control. And you can easily tell that ADO-treated homogenate induced less pathology than the control one, which you can also see in this quantification. So based on these results, we were confident that we did reduce pathological relevant A-beta assemblies before amyloid phase. So based on these results, we were then interested how this early intervention would affect the pathology later in life. So would this early intervention actually have an effect? And for this, again, we treated APP23 with the age of 6 months on 5 consecutive days with an antibody shown in this box. And then we let them sit for 6 months and analyze the animals when they reach the age of 12. And in this case, we again wanted to measure the brain A-beta levels, therefore using the two-step extraction method, but we also included histological analysis. So starting with the two-step extraction method, again, like the previous results with the 7.5 months old age APP23, we do not see any reduction in the triton-soluble A-beta, not in A-beta40 nor in A-beta42. But we did see a reduction in 40 and 42. So even after 6 months of incubation, we still can see that we reached in a reduction in formic acid soluble A-beta. So we then performed histological analysis, including the cortical A-beta load, the plaque numbers, and the plaque size. And for all of these three measures, you can tell that treatment with ADO resulted in a beneficial reduction of amyloid pathology in all these three lines. So some of you might have heard from a major and severe side effect of amyloid beta immunotherapy for AD, which is so-called amyloid-related imaging abnormalities. And one of these abnormalities is cerebral amyloid angiopathy. And this is what we then quantified in our animals to see if this would also appear in our treatment. And you can see that we actually see an increase in CIA score for ADO and M266 animals, but this increase is not very pronounced except for these two outlier mice. So we were not very concerned about this. So we could show, based on these results, that treatment with ADO actually reduced amyloid pathology, especially in plaque number. But we were also interested in seeing what this antibody might do to the plaque structure itself. And for this, we used a specific class of dyes, which is called luminescent conjugated oligothiophiles. And you can see this here. And these dyes, they bind to the repeating beta sheet structure of an amyloid. And by binding to an amyloid structure, this flexible backbone, which you can see here, it's then twisted. And depending on the twisting, the spectral signature of this dye could be affected. So thereby we can detect structural differences between amyloids. In our study, we used two different dyes. One is the QFTAA, which has an emission peak of 502 nanometers, which is shown here, and HFTAA, which you see on the left side, with an emission peak at around 588 nanometer here. And these are the emission spectra of the antibodies. And you can already see that M266 and ADO look a little bit different than control and beta-1. And this becomes more pronounced when we calculated the ratio between these two emission peaks, between 502 and 588. And here you can see that both actually ADO and M266 shifted the spectral signature significantly. So it seems that ADO treatment, it's shifting the spectrum more to the diffuse and M266 to more dense plaques. So this was basically the histological findings, but we were also interested in the biochemically. So up to here, I hope you're still with me and that I was able to convince you that there are pathogenic seeds before plaque onset, and the treatment intervention at this early pre-amyloid disease stage is beneficial regarding amyloid pathology. So obviously 20 to 30 minutes, it's not enough to show you all the data, but I'm really eager to show you our efforts to isolate and characterize the early pathogenic amyloid beta seeds. And for this, former lab members established a method that we termed antibody recognition profiling of A beta assemblies, or short ARPA. And I would like to guide you through this method very quickly. So for this, we use a brain homogenate, which contains a variety of A beta assemblies. And these A beta assemblies are then semi-natively separated by size with an agarose gel. And then these different fractions are collected using enzymatic digestion to liberate these A beta assemblies and make it accessible to further analyses, such as immunoprecipitation with amyloid beta targeting antibodies. You can also use these fractions and have a look on the total A beta in it, so you're not relying on A beta targeting antibodies, which we also did. So we use brain homogenate from old ABP23 mice, size fractionated A beta assemblies with agarose gel, and use the fractions for pull down with the antibodies that we also used for immunization of our mice. And here you can see the Western blots. So right down here, this is the total A beta, how it looks in the different fractions. And you can nicely see that ADO, which recognized the very early seeds, did almost exclusively recognize A beta in the high molecular weight fractions. So this is high molecular weight fractions, and down here, so 1 to 3 high molecular weight fractions, 6 and 7 rather low molecular weight fractions. And ADO almost exclusively recognize high molecular weight fractions, whereas M266, for example, almost exclusively targets the low molecular weight fractions. This is also nicely seen in this antibody affinity plot. So these are basically the results that you see on the left-hand side, but quantified. So beta 1 would look like the same, like the total A beta amount, and that's the reason why we don't have any peak anywhere, whereas aducanumab has a straight peak in the high molecular weight fractions. And this shows you that this antibody specifically or preferentially recognize high molecular weight fractions. And so it shows us that aducanumab, as expected, recognized preferentially high molecular weight fractions, high molecular weight A beta assemblies. But it does not tell us much about what it really recognized in the pre-amyloid phase. So I told you that these are 26 months old ABP23 mice, so they're way ahead they're way above plaque onset. But we were interested on what does aducanumab recognize before plaque onset. And for this, we used 6 months old ABP23 mice homogenate. So this is homogenate which was not treated with any antibody. And we also size separated the A beta assemblies and used these antibodies to pull down A beta from the fractions. And interestingly, we did not detect anything with aducanumab, which did recognize something in vivo, as I hopefully could convincingly show you before. We also did not recognize anything with ME8, which was expected, as this antibody recognized pyrrolidamate, which is a modification which appears quite late in amyloid pathology. So we were really surprised, and therefore we performed immunoprecipitation with these antibodies on unfractionated homogenate, so not size fractionated. And here we can see a very faint band for ADO. So therefore we concluded that that it's most likely that we did not see a band here due to limited analytic sensitivity of this assay. So since this was our limitation, we were still interested whether we would see a difference in the treated animals. So we could see with the formic acid extraction that we in fact reduced triton insoluble A beta. So we were interested whether this would also be high molecular weight A beta. And therefore, we used 7.5 months old ABP23 homogenate from ADU or control-treated animals and subjected this to our ARPA assay. And I think you can nicely recognize that in the ADU-treated animals, we basically lose signals in the high molecular weight fractions of this ARPA, whereas we see something in the control animals. And you can not only see this on the Western blot, but you can also see this nicely in this ratio that we made. So with this, I will already come to my summary. So based on these results, we concluded that there are therapeutically targetable pathogenic A beta seeds before the amyloid formation becomes detectable. And that reduction of early A beta seeds has a long-lasting effects on associated pathology later in life, as shown with the 12 months mice. And this might be necessary to achieve primary prevention for Alzheimer's disease. We also concluded that increasing the analytic sensitivity of ARPA might help to determine pathogenic assemblies that currently escape detection, and thereby we could newly define the preclinical phase of AD. Unfortunately, the structure and biochemical nature of pre-amyloid seeds remain unknown, but they might have different properties compared to end-stage seed. And this is basically also what I'm working on right now in my project. So I'm really curious if I can tell you in, I don't know, next year, another paper. So with this, I would like to thank all my colleagues, my collaboration partner, the authors, especially Matthias Jucker, my supervisor, and Ruth Uhlmann, the first shared author of this work, and you for your attention. And so this is Tübingen. It's a very beautiful small city, and it's not only beautiful, but it also has really good scientific or science-related institutes. And I can highly recommend, if you would like to go aboard or if you want to come to Germany, considering coming to Tübingen for your postdoc or for your PhD. And with this, I'm happy to take any questions. Thank you.
Okay, that was great. Thank you very much. Thank you, Christine. It was a lot of work. so now it's we're open questions to the audience. So everyone can unmute yourself, or you can type your questions in the chat. So feel free to ask questions. So maybe I can start with an experimental design question. So your work is a long-term experiments. You treat the mice and wait for several months, and you harvest the samples at 12 months old. And so I was just wondering, how do you determine the dose, the concentration, and the time point in your experiments? Because I assume that you may not have the time to try and error. You wait for a year and come back to change the paradigm of your work. So I'm just wondering what's it based on, and how did you decide the procedure?
Yeah, so to be honest, I wasn't in the laboratory at this time when the first treatment was decided. But I think, so the first treatment was from 6 to 7.5 months. And this was, so the important thing hereby is that we wanted to start the treatment before the plaque onset. And we also wanted to stop the treatment before plaque onset. So this was the first idea of the experiment. And therefore we, based on what we measured in this early, or in these young animals, we decided to start with 6 and then stop before 8. So that's the reason why we chose 7.5. So this was this. For the long term, like the 6 months, I think this was just educational guess, I would say. So we know that plaque onset will start at 8 months. So we see already a lot of pathology with 12 months. So we wanted to actually go above this plaque deposition time point, but we didn't want to wait until the end stage, because we were afraid that we would lose the effect that we might see. So that's the reason why we chose 1.5 in 6 months.
Yeah, I see. Thank you. I have another question, but I think I should go through the attendees' questions first. You can look at them in the chat. So the first one is, do these antibodies prevent ASC cross-seeds also? I think she's referring to maybe Dr. Hannica's work, the microglial ASC specs, cross-seeds, A beta.
I don't know about cross-seeds. So we haven't tested this, to be honest. I don't know. Sorry,
Okay.
I cannot answer this one.
Okay. And the other question is, can you please advise other strategies for anti-seeding in addition to antibodies?
Anti-seeding in addition to antibodies. So I know that there are some different small molecules which should also prevent aggregation. Although I can't tell any small molecule by name, but I know that there should be some available. So at least they showed that this would prevent aggregation in vitro. I'm not sure if they were able to use it in vivo, but I'm pretty sure there's some strategies about this. I'm not sure what the strategies in terms of start of treatment. I would assume I would also start an early treatment. So if you're interested in preventing, then it should be before disease onset. But otherwise, yeah, I hope this answered the question a bit.
And the next question is, so this person say, I don't know if you said it or I missed it. Which is the route of administrations of your antibody?
No, I didn't mention. Yeah, thank you for reminding me. So we used IP injection, so into the intraperitoneum, and we did not know how much antibody would actually cross the blood-brain barrier. So we did some pharmaceutical experiments that I did not show you. So we were able to see that there was antibody. I mean, obviously, we also could detect treatment effects, but we don't know how much is basically lost. We know it's going through, but we don't know how much. And this is also the reason why we chose 5 consecutive days of injection these animals. So we wanted to have a very, very high concentration of A beta and hope that as much as possible would go through the blood-brain barrier. But yeah, this is a valid point. So this is also a big issue for clinical trials, that the antibodies just not cross the blood-brain barrier. Yeah, great.
Exactly.
Great.
Okay, so the last question is related to microglia. Did you analyze microglia activity in any of these treated mice?
Yes, I also did not show this data, but we did. We did a very talented postdoc in our lab, Angelos Cortales, and he was, so we stained microglia, and then he was able to analyze activated microglia. So he was able to distinguish activated from not activated microglia. And we could show that the number of activated microglia actually was decreased in ADO-treated animals, but not with the other antibodies after 6 months of incubation. Like this. Yeah.